Right, hello there, Year 9, and welcome to the second Walking Talking mock. I'm very glad that people found the last one so helpful. You do Walking Talking mocks closer to your exams, but it is actually something that is really good to do before then, just so you know those command words and how to actually answer questions. So glad that was really useful. Hopefully this will be just as useful. I've split it up into two parts. Depending on how many corrections you need to do, obviously take you more or less time. So please make sure you're completing your corrections as we go through. Annotate your answers, your paper, with points as we go through of how to make sure that you answer it correctly next time. So we're going to go through the first part of the paper. And then in tomorrow's lesson, we'll go through the second part of the paper. So the first question was about the structure of the human heart. So it's given you a picture of a human heart. It has labelled the pulmonary artery, and then there is a letter A. So you can probably already work out that it's going to ask you what that letter is. So we're starting off with a very simple question where you just need to be able to identify different structures. It says 1.1, what part of the heart is labelled A? So you have to think to yourself what the different parts of the heart are. The atria, which is the plural for atrium, they're the top chambers. The bottom chambers are the ventricles. So it's not asking if it's left or right, because that's the other way around, so that can cause a bit of confusion. It's just asking what structure it is. So because it's the chamber at the bottom, then we should have ticked ventricle, and that will be worth 1 mark. If you have multiple choice questions where you don't know what the answer is, do obviously leave them, come back to them at the end, but I would definitely suggest ticking any that you think will be the most probable answer if you don't know. Never leave a multiple choice question. Okay, moving on to another multiple choice question. It says, where does the pulmonary artery take blood to? So it's the right hand side, so it's the smaller side, so it's going somewhere pretty close, and the answer to that is the lungs. Now, if you're unsure whether it was the lungs, then have a look at some of the other options. You've got brain, liver, and stomach. You're not taught any of the arteries that go to those ones, apart from the liver one. So it must be the lungs. Remember to also make sure you are ticking the right number of boxes. It does say tick 1 box, it's worth 1 mark. If it was 2 boxes, it would normally be worth 2 marks. So make sure you only tick 1. If you ticked the lungs and another answer, you would get 0 marks. So make sure that you are ticking the right number of boxes. Okay, so the next question is asking you to identify a structure again, but this time it's not a multiple choice question. It's asking you to add it to the diagram. These questions are often easily missed, where students don't realise they have to actually do something. They think that this will be part of another question. So it's very common that these are actually left. So do make sure you read all of the questions carefully. There's actually several valves on this. I have circled 3 of the main ones, which you can see here, and any one of those would be a valve and would be a correct answer. So when you're looking at valves or thinking about valves, they're normally sort of flaps of tissue that open and close, and they stop the backflow of blood. So those structures fit that description if you didn't already know where they were. So any one of those 3 would get you the mark. Okay, question 1.4. State the name of the blood vessel that brings blood from the legs to the right side of the heart. Now this one, they're giving specific places like legs to try and confuse you. So you need to know from the body where it goes to. So even though it says legs, they could be anywhere from the body apart from the lungs. So state the name of the blood vessel that brings blood from the legs to the heart. So it's going to be a vein. So that's already halfway there. And the one that brings it from the body is going to be the vena cava. Now, if you have put superior or inferior vena cava, because you've learned that in PE, you would still get the marks. But the answer is vena cava for science. Okay, next question. The coronary arteries supply blood to the heart. Figure 2 shows two coronary arteries. So you've got a healthy artery and you've got an artery affected by coronary heart disease. And it labels fatty deposits, the blood flow, so where the blood's going, and the artery wall. Describe two ways the healthy artery is different from the artery affected by coronary heart disease. Now, this question does not actually involve any science that you might have learned in lessons. It is looking at the diagram and talking about two of the differences between the two arteries. It asks you to describe them, so you don't need to say what effect that has or anything like that. You just need to say what the two differences are. So you could say that the artery affected by coronary heart disease has fatty deposits around the outside. The other difference between them would be the amount of blood flow. So you can say that the blood flow is less or reduced or not as much as the healthy artery. Obviously, you might have said the reverse. So you might have said the healthy artery doesn't have any fatty deposits. Or you might have said the healthy artery has bigger blood flow or more blood flow. Either way round is fine, as long as you have spotted the difference of blood flow and the fatty deposit and said which it has. So that will be worth 2 marks, one for the fatty deposit and one for the blood flow. So whenever you're talking about describing, make sure you're not going into too much detail, because that's not going to get you the marks to be able to explain it. You just need to talk about the differences on this one. Okay, 1.6. Suggest 2 risk factors for coronary heart disease. Now, in this question, the command word is suggest. So in this one, they don't expect you to know necessarily the specific risk factors, but we have done risk factors, so we should know 2 of those risk factors. If you have said a diet high in sugar or a diet high in fat, that would be a mark. If you have said poor diet, then that would be a mark. If you've just put diet though, that would not be a mark, because that's not specifically saying whether it's good or bad. If you have said lack of exercise or sedentary lifestyle, or not exercising much, anything like that, that would be a mark. If you again said just exercise, that wouldn't be enough. If you've said smoking or drinking too much alcohol or high levels of stress, all of those would also be a mark. So you would get 1 mark for each of the risk factors that you've identified. Okay, on 1.7 now, we have got a graph. Please be aware that at least 10% of the questions in your biology exams will be math-based. So this would count as a math-based question because you are using graphs. So figure 3 shows the percentages of adults in the UK who have coronary heart disease. Always read the axes. So the y-axis, the one going up, is the percentage of adults who have coronary heart disease. And at the bottom on the x-axis, we have the age groups. So the age groups are 18 to 54, 55 to 64, and then the 65 and overs. There is also a key. So the darker bar, the grey bar, is for males, and the clear bar is for females. The question says, calculate the difference in the percentage of male and female adults aged 65 and over who have coronary heart disease. Now, this is only worth 1 mark, so it is not going to be anything complicated, like looking at the percentage difference. It's just looking at the difference between the numbers. So you need to take the bar in the 65 and over to do with males, which is 18. And then you need to take away the number of females, so 10. So 18 minus 10 is 8. So the correct answer is 8%. In maths, you would have come across the word difference. What's the difference between numbers? It's just simple subtraction. Next question, and I've included the graph on this so we can talk about it and see whether each suggested answer is correct or not. So 1.8, which is the correct conclusion for the data in figure 3? Tick 1 box, and it is worth 1 mark. So we need to tick 1 box. Now, suggest in questions like this, you go through each sentence and decide whether you think that it does do that or not, and then tick the one that you're going to go with. Don't just start reading them, tick the one that you think it might be and not read all of them. Always read every single one. So the first one says, children do not suffer from coronary heart disease. Now, children is anything below 18. So we don't know that because there isn't any data for it. So it's not going to be that statement. The next one says more males suffer from coronary heart disease than females. So if we're looking at those bars, the grey represents the males, and each one, the males are higher. Next one, more younger people suffer from coronary heart disease than older people. So again, we need to look at the heights of the bar. We can see that the younger people, so the 18 to 54, they're much lower compared to the 65 and overs. So it's not going to be that one. So that means that the middle one, more males suffer from coronary heart disease than females, is a correct conclusion we can make from that graph. Okay, so question number 2 now. Biological detergents contain protease enzymes. Figure 4 shows some apparatus and materials. So we've got some test tubes in a rack, we've got a measuring cylinder, we've got a water bath, detergent solution, protein stain, pieces of cloth, and some forceps, tweezers. Describe how you would use the apparatus and materials shown in figure 4 to find the best temperature for removing stains from clothing. You should include how you would make the investigation a fair test. Okay, so let's break this down. First of all, like with the maths, there is a certain percentage that is going to be based on science skills. So that's talking about practical investigations. And it's 15% in all of your exams. So you are guaranteed to come across a question to do with, at least one question, to do with carrying out a practical. It's given the apparatus, but notice that they haven't labelled the test tubes, test rack, measuring cylinder. I would, if this was me, maybe just make a note of that on the paper, just to remind myself to use those words, as well as the ones that have been given to you. Now, there is a required practical on enzymes. Miss Corbett did the video on it a while back. This one is different to that, so please be aware that even though there is a certain practical you have to do, they may ask you a question where it's slightly different. So we've got a bottle of protein stain, we've got some cloth, and we've got some detergent solution. So logically, if we're looking at the best temperature for removing stains, we're going to want to add some stain to the cloth. Now it's asking as well how you would do it as a fair test. So you're probably going to want to mention about how much protein stain you should include, on how many pieces of cloth, how many test tubes you would use for each temperature, how you would use the water bath, and also there's a measuring cylinder, so how much detergent should you use? You will gain more marks if you give specific numbers and amounts of how much you're going to use. So if you say add some protein stain to a bit of cloth, that's not going to get you as many marks as putting add 5 drops of protein stain to 1 piece of cloth. Now this is a 6-mark question, so it is a big question, and we should make sure that we are spending the time answering this correctly. Do not rush 6-marker questions. If you want to make a plan or put some notes down, you are welcome to put that anywhere on your paper. You will not lose marks. So jot down some ideas before you start putting together your sentences. The same as you would in an English exam. If you're asked to write an essay or something like that, then you would have your plan first. Now I've copied onto here the mark scheme, just so that you are aware how the examiner is going to mark it. The first bit that I'm going to point to is the bottom bit, and this is the indicative content. So this is content that they would expect you to have written about. So in here, you said about using a measuring cylinder to measure equal volumes of detergent. So if you have said use the measuring cylinder to measure 10 mils or 20 mils of detergent, that would be brilliant. A lot easier than saying measure equal volumes. You would still have the mark. So if you've written anything about that in your answer, just give it a little tick. It doesn't mean it's worth a mark, but it is something that you have mentioned. The next one that they suggest that you've spoken about is about applying the number of drops of the stain to the cloth. So this is making it valid or a fair test by making sure that the control variables are kept the same. So if you've said put 5 drops or 3 drops or anything like that, an amount of drops of the stain onto the cloth, then that will be a mark. Not a mark, sorry, give that a tick. If you have mentioned about using a cloth with no stains, that also will be good indicative content. So we would use that as a control to compare against. If you've mentioned about using the forceps or tweezers to put the cloth into the test tubes, then that would be worth a mark because it asks you to describe how you would use the equipment. If you've said about in the test tubes putting the detergent solution and the cloth, the stained cloth, give that a little tick. If you've said about using the water bath to give temperatures, the correct temperature, give that a little tick. If you have said, for that one as well actually, if you've said to set the water bath to 10 degrees Celsius, and then later on said 20, 30, 40, etc., then that also is a really good answer. So give that a little tick. If you've mentioned about putting the cloths in the detergent solution at different temperatures, then give that a little tick. If you said about leaving it for the same amount of time, that would be a little tick. If you've said about repeating it at least 3 times, or repeating it 3 times, or repeating it 4 times, anything like that, give that a little tick. And then for the last one, some method of assessing how much the stain has been removed. So you could say about the percentage cover of stain, or you might have said the size of the stain, measure the size of the stain, anything like that, a suitable way of getting our results. So now that you've got your ticks of things that you could have included, we're then going to move to the different levels. So if we start with the top level and work our way down, if you haven't reached the level, then look at the next level and see if you get marks in that. So to get all 6 marks, it needs to be a clear and logical method that you've described. It could be easily followed and would enable valid results to be collected. So somebody else can easily follow it and they'll get the same results as you. That would be worth 5 to 6 marks. So if you have lots of ticks in your indicative content and it is logically ordered, so it's one after the other, then that would be in the top level. So if you've got all of that, that'd be 6. If you've got most of that, then that would be 5. If, however, you don't think that it is part of that level, then we move to level 2. The method described could be followed and would enable some results to be collected. The detail required to ensure valid results may be missing. So if you've got a method, but it's probably not going to get the same results each time it's used by other people. If you haven't said control method, so having the same number of drops or equal volumes of detergent. If you're missing one of those or 2 of those, you can still be in this band. So if you have done that, so if it is reasonably easy to follow and it will get some results, but you have missed a couple of the control variables, you're going to be in this band. So if you've done that 4, if you only put 1 valid control variable, then that will be 3. If, however, it's not going to get any results, like in level 2, then we're going to move down to the next one. So level 1, some steps of the method are given, but they're not clear, they're not logically ordered. Following the method would not give valid results. So if you have not mentioned any of the control variables, you would be in this band. If you have given a semi-useful method. So if you've got bits of the method, but you haven't spoken about any of the valid, the control variables, such as the volume of detergent, number of drops, etc., then you can be in this band. So that will be worth 1 or 2 marks. If you've given no control variables and your method makes no sense, then that would be 0 marks. So this is what the examiner will have, and this is what they will be looking at in order to mark your 6-marker question. There's normally 3 bands for a 6-marker, and 2 if it's a 4-marker. So do make sure that if you are talking about a practical, we are covering repetitions, control variables, pieces of equipment that we're going to use, and that it is in a logical order. You can bullet point for methods. I prefer bullet points, so I would do it as a bullet pointed method. Don't think of it as an essay in terms of one massive paragraph. Just put it into bullet points. It makes it a lot clearer for the examiner. Okay, so moving on to the next one. And I know a lot of people do not enjoy doing graphs, but you are guaranteed to have a question in at least one of your papers where you are going to have to draw a graph. So if you know that that is a weakness of yourself, please make sure that you are practicing and really putting effort into graph drawing in your lessons. So 2.2. In a similar investigation, a student investigated the effect of pH on the time taken to remove a stain from pieces of cloth. So in this one, we've got different pHs, and then it was timing how long it took to get rid of the stain. On the grid below, draw a graph to show the student's results. Add a suitable scale and label to the y-axis. Plot the student's results. Draw a line of best fit. Now, quite often they will tell you the things to include, so I suggest ticking them off as you have done them, because a lot of people forget one of those things, and it will be an easy mark that you can get in the exam. So breakdown, 9 times out of 10, you're going to get a mark for a suitable scale on the axis labelled. So you could do a suitable scale but not include the label, and that could lose you a mark. So make sure you've done both bits. Normally there'll be a couple of marks for the plotting the points, and you have to make sure that you're being as precise as possible. And then there will normally be a mark for the line of best fit. Every time you draw a graph in an exam, make sure you include a line of best fit. And unlike maths, it's not always going to be a straight line. Please remember you can have curves of best fits in science. It's not always going to be a straight line, and never is it going to be dot to dot. Okay, so first of all, apologies for my line of best fit. I did do it on the computer, so it's not like drawing it freehand, so it's pretty wobbly. I have got shaky hands anyway. But this is what I produced on the computer. So let's have a look at the mark scheme that I've included. The y-axis is labelled time taken to remove stain in minutes. I have included that. Plus has a suitable scale. Now, you won't have graph paper as horrible as this. It will be more what you're used to in lessons, where it's got the darker lines on it. Most of the time you'll have that type of graph paper. But I had a look and I counted up the little boxes. I looked at what the biggest result was, which was 20, and tried to get it so that it fitted on. Most of the time they are going to give you the perfect size piece of graph paper. So try and fill it as much as you can. So I've gone up 0, 2, 4, 6, 8, 10, 12, 14, 16, 18, 20. I should have included a 0 at the bottom. Sorry about that. You should have a 0 at the bottom. If you have done that and you've labelled it, then that's going to be a mark. Next one, points or bars plotted correctly to within plus or minus 1 millimeter. Now, this should be a line graph like this, so I'm not going to accept bar graphs from this past paper question, because it shouldn't be, especially if it's asking you to do a line of best fit. You wouldn't often have that with the bar graph. So I've plotted my points. Took quite a while to get them into the right place. So you should have something that looks like this in terms of the crosses. If you have got something that's pretty much like that, then that's going to be worth 2 marks. Notice how it says plus or minus 1 millimeter. So you have to be very, very careful where you're putting your crosses. Please make sure you're being as precise as possible. I used a bit of paper against this graph paper when I made this. So if you're doing the actual exam, use your ruler. Make sure that it's going in the right place. And finally, one suitable line of best fit drawn on the graph. So on this one, it will curve down and then it will make kind of a U shape and then go back up again. So do make sure that you're being careful on those lines. This isn't a great example, but it was after several attempts, the best one that I had on it. Should be a nice smooth line, a smooth curve down, a U, and then going back up again. And that would be your last mark. So well done if you managed to get all of that. If you were doing it on computer, I appreciate how tricky that was. So well done. That's worth 4 marks. Okay, so the last question that we're going to do in this lesson is state the best pH for using the detergent. So we have to think to ourselves, what will be the best time? Is it going to be a long time or a short time? And obviously it's the less time, the shorter the time, the better the detergent. So the cross that was the lowest was pH 6. So well done if you put pH 6. Even if you hadn't done the graph, you could have answered that from the table. So please don't just skip questions anyway. But then even more important, don't skip the whole thing, because you can pick up marks in other places. Every single mark you get is going to be very, very important. It could be the difference between grades. If you are one off that grade boundary, it's going to be really annoying if you could have easily picked up a mark somewhere else. So treat every mark as very important. Right, that is it for me then today. Please make sure that you have done any corrections necessary on your paper, and also add to your revision maps that you've been creating in the previous 4 lessons before you did the questions, just making sure that you have all the necessary information you need. Well done for attempting the questions, well done for doing your corrections, and in tomorrow's lesson we'll go through the second half of the paper. Have a lovely day, and you'll hear from me soon. Take care and goodbye.